Translation: Synthesis of a Polypeptide by Emily McGlame and Ellen Tucker.
Basics. What's involved? During translation, a succession of tRNA molecules add their particular amino acid to the polypeptide chain as the mRNA is moved through the ribosome. The components involved are tRNA, mRNA, amino acids, ribosomes, and rRNA.
In this picture, the process of translation is summarized. A tRNA molecule is attached to an amino acid, GLY in this case, and enters into the A site of the ribosome. It then attaches its anticodon to the complementary codon on the mRNA strand and adds the amino acid to the polypeptide chain in the P site. It then moves to the E site and exits the ribosome to retrieve another amino acid.
What is mRNA? Its actual name is messenger RNA, produced during transcription that carries a genetic message from the DNA to the ribosome in a string of codons. In this picture on the right, you can clearly see the codons, which are 3 nucleotide bases, grouped together like a triplet.
What is tRNA? tRNA stands for transfer RNA, and its function is to transfer amino acids from the cytoplasm to the ribosomes that contains an mRNA strand. This picture on the right shows the two-dimensional structure of tRNA. It is comprised of 4 base-paired regions and 3 loops, including the bottom loop, which contains the anticodon. This anticodon is unique to each tRNA type. At the 3 end, the attachment site for amino acid is located. Since there are only 45 different types of tRNA anticodons, some of them can attach to more than one mRNA codon, allowing them to cover all 61 mRNA codons. The flexible pairing rule that allows them to accomplish this is called wobble. The rules for pairing are relaxed at the 3rd base of a codon and complementary anticodon.
What is rRNA? It is ribosomal RNA, and together with proteins, it comprises ribosomes. It is the most abundant type of RNA because there are several thousand ribosomes in each cell.
The structure of ribosomes. Ribosomes are comprised of a large subunit, a small subunit, E site, P site, A site, an mRNA binding site, and an exit tunnel where the completed polypeptide chain leaves the ribosome. Polyribosomes are also important to translation because they allow multiple ribosomes to translate the same RNA strand at the same time, making multiple copies of the polypeptide simultaneously.
What is amino acyl tRNA synthase? It is a family of enzymes whose function is to correctly match a tRNA molecule to a specific amino acid in the cytoplasm. The active site of each synthase fits only a particular set of amino acids and tRNA. This diagram shows an amino acyl tRNA synthase joining a specific amino acid to tRNA molecule. First, the active site binds to the amino acid as well as to ATP. Then ATP loses 2 phosphate groups, leaving AMP to join the amino acid. Next, the particular tRNA bonds covalently to the amino acid, thus displacing the AMP molecule. Finally, the charged tRNA containing an amino acid is released from the amino acyl tRNA synthase. The tRNA molecule is now called an amino acyl tRNA.
The 3 stages of building a polypeptide are initiation, elongation, and termination. This picture highlights the initiation of translation. First, the small subunit of the ribosome binds to a strand of mRNA. Then, an initiator tRNA with the anticodon UAC base pairs with AUG, the start codon on the mRNA. This UAC triplet is the anticodon that specifies for the amino acid methionine. Following this pairing, the large subunit arrives and the tRNA is put into the P site. Initiation factors, which are not shown, bring together the units of the initiation complex.
During elongation, amino acids are added to the polypeptide chain one by one. In the first step, codon recognition occurs. This is when the anticodon of an incoming tRNA base pairs with the complementary codon in the A site. Hydrolysis of GTP helps to make the recognition process more accurate. In the next step, peptide bond formation occurs. This is when rRNA molecules help form peptide bonds between the amino acid and the A site by removing the polypeptide from tRNA in the P site and attaching it to tRNA in the A site with the amino acid attached. Then translocation occurs. This requires the hydrolysis of GTP once more, causing the tRNA from the A site to translocate to the P site, and the tRNA in the P site, which no longer has an amino acid attached, to exit through the E site. This allows the ribosome to be ready to receive the next tRNA in its A site. Elongation factors, not shown, also assist in the process of elongation.
In termination, hydrolysis of GTP as well as protein factors aid in releasing the finished polypeptide and disassembling the components of the ribosome. First, when a stop codon appears on the mRNA strand, a release factor, which is shaped like a tRNA molecule, enters the A site. The three stop codons are UAG, UAA, and UGA. This release factor promotes hydrolysis of the bond between the last amino acid of the polypeptide chain and the tRNA in the P site. Once the polypeptide chain breaks loose of the tRNA, it exits through the exit tunnel of the ribosome.
Following termination of translation, a polypeptide may require certain processing to create a functional protein. The polypeptide chain might need coiling to form a three-dimensional molecule with secondary and tertiary structure. Often, a chaperone protein is needed to assist the polypeptide during its folding. Post-translational modifications may also occur after the termination of translation. These are additional steps, such as chemical modifications of amino acids and enzymatic cleavage of certain sections of the polypeptide chain.
Targeting the functional protein. During this process, a signal recognition protein, or an SRP, attaches to the signal peptide, which causes polypeptide synthesis to stop momentarily. The SRP then binds to a receptor protein on the ER membrane and leaves when the signal cleaving enzyme on the receptor signals its release. When the SRP leaves, polypeptide synthesis resumes and starts to go into the ER lumen, with the signal peptide still in the receptor protein. The signal cleaving enzyme then cuts off the signal peptide, and the remaining peptide folds into its last conformation and completely enters the lumen. Here is a video that summarizes the process of translation.
Time. When the RNA copy is complete, it sneaks out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Here is our works cited.